Hello, everyone. My name is Davin Wallace, and I'm an associate professor in the School of Ocean Science and Engineering at the University of Southern Mississippi. So today I'm going to be talking to you about short-lived radioisotopes and some field sampling strategies. Before I get started, I want to thank the organizers of IGCP 725 for the invitation to come speak to you today about this topic. So the outline and objectives for this talk include selecting the applicable radionuclides, understanding your field site and as it relates to radionuclide application, sampling your field site, and then a summary of everything that we've talked about. So the first step in selecting an applicable radionuclide is understanding the effective dating range. So I'm going to focus on three radioisotopes today: beryllium-7, cesium-137, and lead-210. I want to note that I'm just very briefly going to describe the effective dating ranges of these, and for any lab techniques and principles associated with these radioisotopes, please watch Dr. Reed Corbett's talk on the topic. So beryllium-7 has an effective dating range of approximately 1 year. Cesium-137 can date between the years 1954 and 1963 CE, and that depends a lot on where you are in the world. Lead-210 has an effective dating range of about 100 to 150 years. So very briefly, all of these radioisotopes generally fall down from the atmosphere, where they become incorporated into the sediment that we are trying to date the coastal deposits associated with. So a couple of other questions as it relates to selecting applicable radionuclides include: what timescales are you interested in for your project? So is it annual? Is it decadal? Is it multi-decadal? So obviously, with the effective dating ranges that I just highlighted, one would select one that would cover the timescale that you're interested in. Are you examining a specific event? Did that event happen really recently? Or did it happen, decades ago? And using that information will help you determine the appropriate applicable radionuclide. Also, this one's a little bit more tricky, but it's nonetheless important. And what are the general sedimentation rates in your field site? And so I realize that that information is not always available. It's probably one of the reasons you're trying to use radionuclides. But it is important to try to get as much information as possible, pre-existing information, because this will really inform you how deep that you need to sample. So do you need to sample very deep and do some sediment coring? Is it mostly superficial? Like, is it a very recent deposit? Can you get at it with surface samples and or shallower trenches? Or is it a mix of the two? Other things to consider include the types of sediment that you're working with. So in general, all of these radioisotopes, when they rain down, they're incorporated into the sort of fine-grained organic-rich sediments. And so in general, there's exceptions to all of this, but in general, the less ideal sediments would be your sort of very coarse material where there's very high porosity, all the way to the most ideal, which would be fine-grained organic rich deposits. So in this case, peat. And we know as geoscientists that there's a wide range of sediment types in between. So that's a good rule of thumb. And again, there are exceptions. Another important thing in working with a particular field site is understanding areas of erosion and deposition. Again, I realize that this may not always be known, but in general, it's good to avoid areas of erosion and select depositional sites. Another important aspect to this is to recover enough material. And in general, so the detectors that I use in my lab, we need approximately 20, 25 grams of dry weight of sediment. And that also is going to vary, in the field if you're collecting sand, mud, or peat, the water content after, drying is going to have a major impact on what the dry weight is going to be. So make sure that you recover enough material to do this analysis. Sometimes that means collecting multiple cores or multiple samples from a trench, for example, of a particular layer or layers below the layer of interest. Another important aspect is to capture a spatial distribution. So, either transects or, several samples around a particular deposit of interest or site of interest, because as I'm going to show you, there can be quite a bit of variability when applying these techniques. So this is a meta-analysis of cesium-137 and lead-210 around the world. And I think it illustrates an interesting point, which is that, first of all, as time has gone on, so from 1977 to 1990, there were a few amount of studies. From 1990 to 2005, the amount of studies picked up. And then to present time, there's lots of studies. And if you look at everything together, you can see that there's quite a bit of information about cesium-137 and lead-210, which is often coupled together for a sedimentation rate estimation. There's quite a bit of studies around the world and in coastal zones. And this is important to note because you can look at prior information that might be available in your particular site of interest. It will also, maybe help determine the concentrations that you're looking for these particular radioisotopes, which is important in terms of how much material you would actually need to conduct the analysis. But also it can tell you about the sedimentation rates and thus how deep you need to sample to use a particular radioisotope, as well as the sedimentary environment and the dynamics that are associated with the preservation of high radioisotope concentrations. And so looking for prior information in a lot of these particular areas can be the very first thing to do, really, when trying to understand your particular field site. One of the most important aspects of understanding the application of radioisotopes for a particular site is that there can be quite a bit of variability. This is an example from the Pyrenees in Spain. And so I realize that, that's not necessarily the environment that many of you are looking at, but I think this illustrates a really important and fundamental point that there can be a lot of variability. And so this is just showing cesium-137 and lead-210. And what's also shown is the organic content, the inorganic content, and the clay percentage, as well as the sand content and the concentration of these isotopes. So in the colored map, you can see that the concentration of both cesium and lead radioisotopes is low in the red colors and high in the blue colors. And so you can just see from this map that there's quite a bit of variability in one study area for where you have high and where you have low concentrations. And obviously, the higher the concentrations, the better it is for developing radioisotopes as a dating technique. And so another important aspect is that not only is there variability, but the reason for that variability. So in this particular case, you can see that there's quite a bit of relief across transects that you can see, a little bit over 100 meters of relief. So that's an important aspect because, as you recall, zones of erosion and zones of deposition are important aspects for identifying the applicability of a radionuclide. But there are additional aspects that I mentioned to you before that you can kind of see a little bit more quantified. You have organic carbon here, inorganic carbon, as well as clay percentage and sand percentage. And all of those in tandem are going to play a pretty big role in essentially what ultimately the concentrations are of, in this case, cesium-137 and lead-210 at any given location along these transects. And so you can imagine, depending on your particular site that you're looking at, this highlights, again, the importance of trying to find as much information about the site as possible in terms of what the concentrations look like, what's the spatial variability. And if there's no prior or pre-existing information about this again just highlights the need to really get a good spatial distribution and maybe even different sediment types above and below your layer or layers of interest, because there are going to be these complex relationships that are not always easy to quantify. So in summary, this is a general order, but some of these things are happening, in tandem. They're not necessarily, happening in this serial order that I'm presenting, but this is a general understanding of the field applications of radionuclides. So first, select a radionuclide and keep in mind the time scale of interest that you all have. So is it an event scale? Is it decadal? Is it multi-decadal, et cetera? Then ask yourself, is the appropriate sediment type for the preservation of these radionuclides essentially, is the appropriate sediment type present? So fine-grained, organic rich in general tends to be the best, not always, but having something like that is usually a good bet. The next thing is selecting areas of deposition and avoiding areas of erosion. Again, as I said, I know that's not always possible, but if there's anything obvious that you can see that would help you to delineate that aside from the pre-existing studies, use that information and try to select areas of deposition. Finally, when you've gone through all of that, sample deep enough to capture the event and the events of interest that is applicable to the radionuclide that you're trying to capture. So this might mean if it's very deep, if you have high sedimentation rates, then you need to get longer cores. Could be that you're looking at an event that just happened, and so that would be just at the surface. And so just make sure that one sample is deep enough to actually capture what you're looking for, and maybe even go a little bit below it if you can, just to be on the safe side. Because there are some assumptions that are made. If you think an event is something and it turns out to be something else, it's always good to have the most amount of sediment and the deepest record that you can reasonably capture. Next, make sure that you recover enough material. And so, as I said, the detectors that I'm familiar with and the ones that I use are typically the best results are using about 25 grams of dry weight. And so obviously that depends a lot on sediment type. So if you have very water-rich sediments, obviously, when you dry them, they're not going to be very, very large dry masses. And so make sure that you recover enough material. So additional cores or additional samples that you get to that approximate weight. And that does vary quite a bit. But I think it's always good practice if you can to get as much sediment as possible. And finally, it's always good to sample a spatial distribution. So this can help quantify and understand the variability that might exist at your site. It can also help understand areas of erosion and deposition we just previously talked about. And so it can also tell you about bioturbation, which is something that we're trying to avoid with these dating techniques. I want to thank the organizers for the invitation to present, and I hope to talk to many of the attendees in the breakout session. Thank you.